Everybody, my name is Jose Ortega. I am a second year master student in the Riggs Lab here at SFSU, and I would like to preface my talk by asking you guys a question that I'm frequently asked, which is, what do fruit flies and humans have in common? And a lot of people ask me that question when I explain to them that I'm in a fruit fly research lab. And my response to that question is twofold. The first response is that I tell people is that 75% of genes that cause disease in humans are also found in fruit flies, which is pretty interesting to think how something so small like a fruit fly is something so big like a human being have the same or similar genes found within their genome. The second thing that I like to tell people is that they undergo two types of cell division. The first type of cell division, which is shown here, is called symmetric cell division, which is when a mother cell divides into two identical daughter cells. The second type of cell division is called asymmetric cell division, in which a mother cell divides into two different types of daughter cells. Now, my project is looking at how a cellular component other than the DNA aids in cell cycle progression. Specifically, what I'm looking at is I'm looking at the endoplasmic reticulum, also known abbreviated as the ER, in neural stem cells and how that may aid in cell cycle progression. Now, the basis of my project stems from preliminary data found in my lab, which showed that when the ER is disrupted or mutated in a neural stem cell, it causes two types of mitotic defects. The first type of mitotic defect is the neural stem cell is able to reach a metaphase of mitosis, but then takes a very, very long time to divide. We dub this mutation mitotic delay. The other type of mitotic defect was that when the ER was mutated in the neural stem cell was able to reach metaphase, but then once it reached metaphase, never divided. So we call this mutation mitotic arrest. Now, what was interesting about these mutations is that the cells were able to reach metaphase, which is a fundamental step in mitosis. What's known about metaphase is that the spindle assembly checkpoint gets activated when the cell senses that the DNA is not bound to microtubules. However, what's interesting about my project is that when we mutate an organelle, which is not DNA, it also seems to activate the spindle assembly checkpoint. Therefore, my project is to look at what happens when the if the ER interacts with the spindle assembly checkpoint to not only aid in mitotic progression, but also properly inherit the ER into the two daughter cells. So at the end of my term here at SFSU, my goal is to add another thing to when I tell people what do fruit flies and humans have in common, which is not only to explain that they have the same or similar genes, they undergo the same type of cell division, but they also inherit the ER in a similar or same fashion, and the ER aids in mitotic progression in the different cell types found in both humans and in fruit flies. Thank you.